Okay, here we go. We've got oxidative phosphorylation and chemiosmosis, which is the electron transport chain, kind of all in one. Remember, we have NADH, we have 10 of those, and 2 FADH2s, accounting for most of the energy extracted from your food items. Of course, we'll talk about later in the last two sections how you can take lipids and proteins and do the same thing. The two electron carriers are going to go now, and the electrons, of course, from glycolysis, the NADH from a glycolysis, and the NADH from pyruvate oxidation are going to go into the electron transport chain, oxidative phosphorylation, and that's going to power ATP synthesis, which is the chemoosmosis part. Okay, so the electron transport chain is happening on the inner membrane or the cristae of the mitochondria, and it is a series of proteins. They look a lot like the phospholipid bilayer. I love figure 9.15 because it does look like the actual phospholipid bilayer, but this is the inside. You can kind of see the arrangement on page 175 of what the cristae looks like. So you can see there is, when we start talking about the intermembrane space versus the mitochondrial matrix, what we're talking about. The carriers can alternate. They alternate between being reduced and oxidized as they accept and donate electrons. So what that means is that NADH will then go back down to being NAD plus, and that energy electron, the electrons are going to flip-flop back and forth between all the proteins. The electrons drop in free energy all the way to the very end, where oxygen is the acceptor. And ironically, it's kind of weird, but the oxygen accepts it and forms water. So you're like, now wait a minute. So we're going to pass all these electrons, but then we're just going to get water out of this? Well, actually not. As we go through, we're actually going to be pumping these proteins are going to actually be pumping hydrogen ions, and that difference, so that's the proton pump we were talking about earlier in chapter 7. When they pump those protons, we're actually going to get a difference in charge and a difference in pH difference between the inner membrane and the inner membrane space in the mitochondrial matrix. So here shows you the different complexes. What I want to point out here too, so this is the free energy, so that they want you to see as the free energy goes down. The pull of the oxygen down here is really what pulls it down, because oxygen is so electronegative compared to the rest. And here's the different complexes. Notice we've got, so this is talking about the different ones we've got. I wanted to give you the actual name, flavin. Oh, let's see. Flavin mononucleotide. You've got this one with the Fe. You've got different carriers here too besides these multi-protein complexes. You also have cytocrines that go along the cytochromes. Most of the remaining electron carriers between the coenzyme Q, which is right here, and the oxygen at the end are cytochromes. Now, I don't know if you remember what cytochrome means, or if you remember hearing that word, but we looked at cytochrome C in freshman biology, and we were looking across. It was that big grid where we were seeing what amino acids are different in the cytochrome C actual complex between all species of organisms. And of course, the ones that are most related or closer related had the least amount of differences, for example. So you've got all kinds of things as we're passing around. Something else to point out, so we have NADH goes to NAD plus. Okay, as that goes on, it becomes, we're actually taking that away. Also note that we have the FADH2 jumps in at this level. So kind of weird, you actually are going to produce less energy using FADH2 than you would with the NADHs. Which is kind of interesting. So we want to look at why maybe that FAD is involved, because they're going to jump in at step 2, not in step 1. So they're not going to give off as many electrons on that as they would in the other. So we're going to keep going and flip-flopping back and forth. So it's going to gain all 10 NADHs and the 2 FADs as it goes along the way. And again, at the end, once you accept those electrons at the end, we're going to take those electrons We're going to take a half of oxygen, so just one O, add the 2 hydrogens to it, and make water, which is a byproduct of cellular respiration. But you might be asking, okay, well, what happens to all those electrons as we go through them? So one more time, going to the cytochromes, for example, are the proteins, including cytochromes. They have ions all the way to the oxygen, so as you're passing them along. Again, the big proteins are going to be proton pumps. So as the energy is passed on, those are actually going to transfer hydrogens to the intermembrane space. Okay, so it transfers it from the mitochondrial matrix to the intermembrane space across there when we transfer those electrons. We have I have not mentioned at all any ATP. There is no ATP being used here. Okay. Now, once you get to the end, so we've got the oxygen, once you get to the end, the electron transfers, so you cause it to pump hydrogens from the matrix, the mitochondrial matrix to the intermembrane space. The hydrogens are now going to move back across the membrane through a proton, so ATP synthase. And this is actually, there's a picture here that they show. It's actually it's interesting. It looks very much like a motor. They actually give these names, like there's a rotor, like it actually is a turn that actually happens. And there's like a catalytic, now I kind of think about it. I have no idea on cars, but like a catalytic converter, I'm not sure if that's what happens. But as the hydrogens come in and bond, then the turn, that actually generates, we have ADP goes to ATP. So we're actually pumping it. And here's the cool thing. Number one, you don't expel any energy because here's your high concentration of hydrogen and we have a low concentration of hydrogen here. So we are actually going according to the concentration gradient from high to low to make the ATP. So you don't have to expel any ATP to actually make the ATP, which I think is pretty cool. Also, you can kind of think as an addition of lots of hydrogens, there is a pH difference. pH is a measure of how many hydrogen ions that you have. So the more hydrogen ions you have out here, the lower the pH is. So like hydrochloric acid has lots of H pluses, and inside here would have a higher pH, so that you could think about hydrogens as in a pH difference. Here showing you as the NAD, here's your carriers going along here, okay, as they go through here. Notice where Q is in the middle of here of these hydrophobic region. So as it's carrying along, this is causing these guys to pump hydrogens out through here. And at the very last complex, so notice this one is interbed. This is on the inside. I kind of thought that was a transmembrane again. Another one pumping across. So notice this guy doesn't necessarily pump across, just these guys that are the intermembrane ones, or transverse membrane, I guess is what you could say. Making the water here. And then here we are. The hydrogens are coming back inside again. I guess I wouldn't say inside, but that's going into the mitochondrial space. And here I'm making ATP. That is chemiosmosis. Okay, chemiosmosis. Now, something to think about. We're going to talk about anaerobic and fermentation in another slide. Okay, and we'll start it tomorrow. At this point, okay, and this is one of the questions at the end. Wonder if you didn't have oxygen, okay, as an acceptor here. Number one, if you didn't have in the presence of oxygen, if you didn't have that pull, it would stop. Oxidative phosphorylation is not oxidative phosphorylation if you don't have oxygen at the end, okay? So if you don't have oxidative phosphorylation at the end, that's just, it's going to stop completely. One of the other questions, wonder if you had the outside, it says number 2, in the absence of oxygen, what do you think would happen if the pH decreased in the intermembrane space, which is out here? So wonder if you had, so if the pH is decreased, that means there's more H's. So then ATP is going to be made even better because you're flowing from the concentration gradient. And then number 3, in concept 7.1, learn that membranes must remain fluid. Remember we talked about how it cools? So how would the operation of electron transport if the fluidity of this change? Well, you can see Q here has to be able to move. It's in between. So if this doesn't If this is not able to move from here to here to kind of transfer things, then we'd have a major problem. And that whole idea of H plus gradient, having a higher gradient on the outside than that, is called proton motive force, which I love that word, emphasizing its capacity to do work, that you can actually do work because there's a concentration gradient difference, which I think is really cool. And the accounting for the ATP production. So during cellular respiration, again, glucose to NADH to the electron transport chain, proton motive force gives us ATP. That's 34% of the energy in the glucose molecule is transferred to ATP, and you can make about 32. That's awesome, because they mention in here even the best, most efficient automobile actually only converts 25% of the energy stored in gasoline. So I think that's a super efficient number. Now, there are several reasons why we don't necessarily know why there's ATP exactly, like how much ATP that you get, somewhere around 30 to 32 ATP. And part of that has to do with the shuttling part. Okay, so we want to talk about, so one more time, I think this graph or this picture shows some great examples of looking at, okay, again, the big picture. 2 ATP here, we got 2 ATP, that's 2 net gain. Remember, we used 2, we got 4, so we get 2. And here you get 26 to 28 ATP, so around 30 to 32 total. So part of the reasons here, number 1, they gave you 3 reasons. Okay, so one of the reasons I wanted to see, the first reason, phosphorylation and redox reactions are not directly coupled to each other. So the ratio of the number of NADH molecules to the number of ATP molecules might not be a whole number. Meaning this, 1 NADH means 10 hydrogen plus being transported out of the inner mitochondrial membrane, but the exact number that re-enter might not be the same to generate 1 ATP. Like, okay, you might need 10, but it might not actually be that accurate. For example, they now look at, therefore, like a single molecule of NADH, for example, generates enough proton motive force for the synthesis of 2.5 ATP. It's not necessarily energy in and the same energy comes out. Okay? So you might get 1 NADH, it might not make just 1. It's not a 1-to-1 ratio necessarily. And because FAD only joins on the second step, they actually looked at there might not only be enough hydrogen ions for the synthesis of 1.5 ATP. So the proton pumps might not be pumping out an equal amount of what NADH is coming in. NADH might donate 10, but we might not be getting out 10 hydrogen ions. We might be getting less or we might be getting more. We don't know what that generating force is. Number 2, the shuttle. We don't know how much ATP is being used or what's going on when we have to shuttle things, like the 2 NADHs that were out in the cytosol and getting them through and actually to the electron transport chain. So they talked about how it might be different in different types of cells in the body. It just depends. They talked about brain cells and liver cells being a little bit different. So we might be getting out different. It might not be a whole number of ATP. And the third variable they talked about, it might drive other work. So it says, for example, proton motive forces powers the mitochondrial uptake of pyruvate from cytosol. So we don't know if that's being used in other parts that we haven't talked about. But we do know this, and this is what I want you to keep in mind. We know that in the presence of oxygen, we definitely have a bigger production of ATP versus substrate level phosphorylation. The last part of page 176 to 177 talks and throws a lot of numbers out as far as how much energy is created by one glucose molecule and how much you're storing and things like that. But I keep in mind the whole 32% is pretty huge. 32% of that glucose. So about a third of that glucose is being harnessed. So 2/3, that just gives you the whole thing. 2/3 of that glucose molecule is released as waste product, which I think is really interesting. But it's more than, say, if you were to just burn. If you're starting a fireplace, you're burning all that heat. At least you're capturing some of that heat to do work within the cell. Okay. The last part, they also talk about storing energy, and they talk about brown fat, and we'll talk about that tomorrow, and how that's used to actually help lower body temperature yet keep things working. So we'll talk about that tomorrow. But giving you an overall range of the electron transport chain, and we'll talk about the brown fat tomorrow and get started on fermentation and aerobic respiration.